Hello, everybody. Thank you for joining today's webinar. My name is Jasmine Trigg, and I will be discussing live cell imaging strategies to quantify phagocytosis. In this technique talk, after a quick introduction into live cell analysis and kinetic monitoring, I will cover the following objectives. So I'll provide an overview of validated assays for image-based fluorescence readouts of phagocytosis, and I'll show you how these can be easily implemented into your development workflows. I will also provide some guidance on experimental setup and optimization of these assays, and then I'll exemplify their use across a range of research areas. So I really hope that you find the next 30 minutes interesting and also useful. Phagocytosis, a specific form of cellular endocytosis, is a key mechanism by which microorganisms, such as pathogens, and unwanted cells, such as foreign or dying ones, are removed from the body. It involves a complex set of processes, which I've illustrated here in the schematic on the right. So this involves the target being recognized by the phagocyte and engulfed. We then have a phagosome formation, and followed by the fusion of lysosomes to form a phagolysosome. We have the release of lysosomal enzymes, and these result in the degradation of the target, followed by the eventual release of soluble debris. Phagocytosis is a critical component of both innate and adaptive immune responses, and it is performed by a number of professional immune cells, such as macrophages, neutrophils, microglia, and immature dendritic cells. Current approaches, such as high-content analysis or flow cytometry, rely on endpoint assays that provide limited kinetic information and do not allow us to study any cell morphological changes that are occurring. Live cell analysis methods developed using pH-sensitive probes, such as Frodo, offer an alternative kinetic, image-based assessment of these biological events in real time. So what is live cell analysis? In simplistic terms, it is continuous cellular measurement. Live cell analysis using specialized systems, such as the Incucyte shown here, allow us to monitor all research phases, from checking cells or monitoring their manipulation prior to the assay, to visualizing them all throughout the assay time course. Additionally, this instrument is flexible, and this can answer multiple questions, for example, morphology, health, or function. This illustrates application across a whole workflow that provides relevant information in real time, enabling data acquisition of living cells in a non-perturbing manner and for up to long durations of time. The data I'm about to show you has been acquired using the Incucyte live cell analysis system, which is an integrated solution of hardware, software, reagents, and applications, all designed to work together. The hardware is a microscope optimized to permanently reside inside a standard lab incubator, and it can take images from cells cultured in a number of different types of vessels. So you can connect to the Incucyte from any location using the software, which allows you to remotely schedule scans or look at your data in real time. We also provide a suite of non-perturbing reagents and consumables, which allow you to run these assays. And the box at the bottom here on the right illustrates some of the applications that we have that are relevant across a number of different biological areas of research. The Incucyte phagocytosis assay combines Frodo for Incucyte reagents and the Incucyte live cell analysis system for image-based fluorescence measurements of phagocytosis. So if we look at the assay principle, Frodo reagents for Incucyte are a pH-sensitive conjugated fluorophore that has been incorporated to enable live cell imaging of phagocytic events. The principle is based on a low level of fluorescence of Frodo-labeled material in media at around pH 7.4, which, following engulfment of phagolysosome formation, results in an increase in the acidic environment, and this leads to an increase in fluorescence intensity within the cytoplasm of the cell. Using the Incucyte, this fluorescence can be automatically quantified over time, using integrated software to segment the areas of high intensity with minimal background fluorescence. And this assay format is really flexible. So these protocols are simple mix and read ones, and they're easy to implement into your development workflow. For example, you can use your target and phagocytic effector cells of choice, and these assays can be conducted in a high throughput manner, with the option of building in complexity to model for disease relevance if you so choose. Additionally, as these assays are non-exhaustive, they can be used upstream of any endpoint assays that you'd like to do. For example, looking at activation states of immune cells or checking the cytokine levels in the microenvironment. In this webinar, I will cover 4 applications of the Incucyte phagocytosis assay, all of which use live cell, non-perturbing imaging and analysis. First, I will start with the biparticle phagocytosis application. As I previously mentioned, phagocytosis is a key mechanism by which microorganisms are engulfed and degraded, and this represents a vital facet of the innate immune response to pathogens to defend the host from infection. The Incucyte biparticle phagocytosis assay enables real-time visualization and analysis of phagocytosis of infectious agents, for example, gram-positive or gram-negative bacteria or yeast-derived pathogens by immune cells. And this protocol is a simple mix and read protocol that uses sterile pre-labeled photobioparticles, and these are available in either green or red. So I've got a protocol quick guide here, which is shown for a 96-well plate, but this can be adapted to other formats. So firstly, on day 0, we seed the effector cells, and then these are cultured overnight. The following day, if you are wanting to look at any modulators of phagocytosis, we can treat the effector cells with your desired treatments. And then following their incubation, we can add the bioparticles of choice directly to the plate containing your effector cells. Looking at the tips for this assay, firstly, we recommend optimizing the effector cell density. So we want a seeding density that will result in around 10 to 20% confluence 24 hours after plating. So we found that around 10,000 cells per well is a reasonable starting point, but we recommend optimizing above and below this. It's probably worth noting here that phagocyte cell growth can be monitored using the Incucyte and our confluence algorithms. The second tip here is to optimize the concentration of bioparticles. And then the third tip is probably the most important one, I would say. So we kind of strongly recommend sonicating and vortexing the bioparticles in a glass vial prior to the addition. And this is to ensure that you don't get any clumps of bioparticles in the assay plate. And lastly, is to acquire the scans in the Incucyte immediately. So the phagocytosis, especially for the bioparticles engulfment, happens quite rapidly. And so we want to make sure that we capture this as soon as possible. So before I show you some of the bioparticle phagocytosis case study data, I thought I would do a very quick introduction to the Incucyte software. This slide highlights the simplicity of the Incucyte software and its easy-to-use guided interface, from data acquisition to data analysis and data visualization. Automated tools provide a setup and walkaway experience so that you can track phagocytosis in real time. For phagocytosis data acquisition, we use the settings shown on the top right here. So as I've said before, we want to acquire these scans immediately after plating, and we have quite high frequency scans for this assay as well. So we recommend repeat scanning every 15 to 30 minutes for up to 48 hours or the duration of your assay. These assays use a standard scan type and the phase and a fluorescence image channel. And this fluorescence image channel will depend on the Frodo reagent that you're using. We suggest using a 10x or 20x objective and a scan pattern of 2 to 4 images per well. And the objective and scan pattern will depend on the cells that you're using in the assay. For phagocytosis data analysis, as effector cells engulf target cells, the area of fluorescence and intensity inside the effector cell increases. This can be reported in 2 ways. Either as a quantification of an increase in fluorescence area, and for this we use the total fluorescence object area metric, or as a quantification of an increase in intensity integrated over the area of detectable fluorescence. And for this we can use the total fluorescence object integrated intensity metric. Here we demonstrate how the phagocytosis of Frodo E. coli bioparticles by mouse macrophages can be monitored and quantified over time. Shown are the acquired images at 0 hours on the top row or 12 hours at the bottom, and the fluorescence segmentation mask shown in pink that was used to quantify an increase following bioparticle engulfment. These images also highlight the minimal background signal of Frodo reagents. This method can robustly be used to study phagocytosis and its pharmacological modulation in a high throughput format. To exemplify this, we examined the phagocytic capability of rested or polarized iPSC microglia to a density range of bioparticles or labeled apoptotic cells, or looked at the inhibition of engulfment using cytoclasin D. And we did this all within a single 384 well plate. The microplate view shows the kinetic change in fluorescence area for all wells over 24 hours, with varying profiles for the different conditions being observed. As discussed, the bioparticle phagocytosis assay involves the addition of pre-labeled target materials. For the next 3 applications that I'll guide you through, these involve an additional step of labeling your target cell or material of choice. So before going into these applications in detail, I'll first describe the Frodo labeling steps and the initial labeling optimization experiments that we recommend. So the process for labeling your target cells or material of choice is straightforward. And the Frodo cell labeling kit, shown here on the top right, comes with the 4 components that are needed for this labeling. So the kit contains a wash buffer, a labeling buffer, the Frodo dye, for labeling, and also some DMSO to create your stock. So shown here is a brief overview of the target cell labeling steps. So firstly, we want to harvest our target cells and wash these using the wash buffer. Next, we then want to harvest them again and resuspend in a labeling buffer. The third step is the Frodo labeling. So we want to add the Frodo labeling dye at an optimized concentration to the target cell suspension, and we incubate this for 1 hour. And then following the incubation period, we then remove the excess Frodo dye from the cells by washing this in the target cell media, and then we can resuspend the cells again in media ready for the assay. So the Frodo labeling concentration is cell type dependent, and it is very important that the target cells are efficiently labeled with Frodo in order to detect phagocytic events, and we have an optimization protocol for this. So here I'll briefly take you through the target cell labeling optimization experiment. So for this, we recommend performing a serial dilution of the Frodo cell labeling dye for Incucyte in DMSO and labeling the target cells at each concentration. And an example plate map for cultured cell lines for this is shown here on the right. Following the labeling of these cells, we can then harvest and wash these cells, and then we add each dilution to media or citrate buffer conditions. And the citrate-based buffer is at pH 4, and this mimics the acidic environment of the phagosome, in which we get that increase in Frodo signal. So these can be aliquoted into a 96 well plate, and once the cells have settled, we can then acquire scans using the Incucyte. If you're optimizing for an Phagocytosis assay, you'll first need to induce apoptosis of the target cells. So after applying an analysis definition with a low red threshold, for example, between 0.5 and 1 red count units, the Frodo intensity can be quantified. So shown here is the mean intensity graph for Frodo red labeled Ramos cells at 2 hours. The solid bars are the media conditions and the open bars are the buffer conditions. And when selecting the Frodo concentration for your target cell, it's important to select a concentration that is easily masked in the buffer condition and has a good signal and exhibits little to no fluorescence in the media only condition. For example, on this graph, we'd recommend suggesting one of the lower concentrations because we've got that lower minimal signal in media, but we've still got a good mean intensity in the buffer conditions. By counting the number of phase and fluorescence objects, we can also get a percentage of labelled cells for each concentration of dye. And as you can see here, across all of the concentrations in this experiment, we had a good labelling efficiency. The percentage of label cells can also be calculated using the cell-by-cell analysis module if you have this.
So now that we've gone through some of the labelling optimization, I will talk you through the phagocytosis application.
The clearance of apoptotic cells, termed phagocytosis, by macrophages is crucial in resolving inflammatory events by the removal of neutrophils that remain following inflammation. Phagocytosis also prevents the development of chronic inflammation in disease and can remove apoptotic tumour cells following anti-cancer treatment. This assay enables quantification of phagocytosis using the Frodo cell labelling kit for Incucyte and your choice of target and phagocytic effector cells. This assay requires you to induce target cell apoptosis, and we recommend conducting a preliminary experiment to assess the lowest concentration of drug treatment, for example camptothecin, that will induce target cell apoptosis with limited cellular debris following a 24-hour exposure. This target cell apoptosis can be measured using the Incucyte and XM5 dyes. It's also important to ensure that experimental design includes replicate wells of each condition being tested, for example, effector cells and apoptotic target cells alone at each density. So shown here is the protocol for this. So on day 0, we want to seed the effector cells, and these can be cultured overnight. On the same day, we can treat the target cells with the apoptosis-inducing reagent. On day 1, we harvest the apoptotic target cells and we label with the Frodo labelling dye, and this will be at the concentration that you would have previously determined. Following the labelling of the target cells, these can then be added into the plate containing the effector cells, and images can be acquired in the Incucyte. So tips for this assay. Firstly is to grow enough effector and target cells in advance. And as part of this is that you want to make sure you're accounting for some loss of apoptotic target cells during the washing steps. We also recommend testing the target to effector ratios to ensure that you get the optimal assay window. And lastly, especially during initial experiments, we suggest using a highly phagocytic cell type as a positive control. For example, the J774As are a highly phagocytic cell line.
So now I'll show some case study data looking at resolution of information. So here we have a high frequency video of phagocytosis that was acquired using the Incucyte live cell analysis system. Mouse macrophages were seeded into an image lock plate co-cultured with Frodo labelled apoptotic Vamos cells, and images were taken every 2 minutes. So we can observe the increase in red fluorescence following the engulfment of apoptotic target cells. These videos highlight the morphological and dynamic insights into phagocytosis that we can gain through live cell imaging.
Cytokines present at the site of inflammation can initiate a cascade of events, such as inducing macrophage polarization to either an M1 pro-inflammatory or M2 anti-inflammatory phenotype. To examine the relationship between these macrophage phenotypes and ephagocytic ability, we use the THP1 cell line. which is an established model of human monocyte function that can be differentiated into macrophages. THV1 monocytes were differentiated to M1 or M2 through cytokine exposure and co-cultured with Frodo-labeled apoptotic Jurkat cells. The phagocytic ability of differentiated macrophages was assessed using live cell imaging, and the fluorescent signal of the engulfed Jurkat cells was quantified. The data revealed a difference in phagocytic capability between polarized macrophages. M2 had a greater phagocytic capability compared to M1 at 16 hours. And this is consistent with the anti-inflammatory function of M2 macrophages in the resolution of inflammation. Next, I will focus on the antibody-dependent phagocytosis, or ADCP, application. Mechanisms exist whereby viable cells can be targeted with pro-phagocytic agents that promote engulfment and clearance through engaging host phagocytes. These mechanisms often involve the addition of an antibody to drive clearance, for example, antibody-dependent cellular phagocytosis, or the blockade of don't eat me signals such as CD47, which are present on the surface of tumour cells and enable invasion of phagocytosis. Pro-phagocytic monoclonal antibodies that target tumour cells hold great therapeutic promise in oncology. The antibody-dependent phagocytosis assay enables quantification of ADCP of target cells by macrophages using the Frodo cell labeling kit for Incucyte. So the target cells in this case should be viable non-apoptotic target cells. Prior to initiating the assay, we recommend that your experimental design includes replicate wells of each condition being tested. For example, labeled target cells alone, as well as target to effector co-cultures at each ratio, in the presence or absence of the antibody, an isotype control or vehicle control. And these controls are really important in determining the optimal assay window for this. The protocol shown here is for ADCP using non-adherent target cells. So on day 0, we can seed the effector cells into the 96-well plate and culture overnight. The following day, we can then label the target cells with the Frodo labeling dye, which concentration has been previously optimized. And then following the labeling of target cells in a separate compound plate, we can combine these with the antibody treatments of choice. Following the incubation with the antibodies, we can then seed this mixture into the plate containing the effector cells. So tips for this assay. So firstly, it's important to ensure the gentle handling of viable cells. So this is all throughout the labeling and also seeding. And this is because we want to be measuring phagocytosis and not epicytosis. Sometimes it's useful to use a hemocytometer and trypan blue to just check the viability of your cells post-labeling. Additionally, in initial experiments, it's often useful to include an apoptotic positive control. And this is really just to confirm that your phagocytic effector cell is capable of phagocytosis. And lastly, we recommend testing the target to effector cell ratios. To assess ADCP, we use 2 clinical monoclonal antibodies targeted against CD20, rituximab and truximab, which is a rituximab biosimilar. And these are both approved treatments for CD20 positive B-cell cancers. Viable Frodo-labeled Ramel cells at a range of densities were treated with anti-CD20 antibodies or IgG isotype control and co-cultured with bone marrow-derived macrophages. Truximab and rituximab resulted in an increase in fluorescence following engulfment of target cells in a time and density-dependent manner. Additionally, treatment with a range of rituximab concentrations revealed that there is a concentration-dependent effect on tumour cell phagocytosis with increasing levels of monoclonal antibody. For all concentrations tested, IgG control had little or no effect on engulfment.
Again, we have some high-frequency videos that were captured using the Incucyte. So bone marrow-derived macrophages were co-cultured with Frodo-labeled Rammel cells in the presence of truxamol isotype control. And we can see this increase in fluorescence following engulfment of viable target cells for truxamol, which is not yet seen in the control conditions.
To investigate don't eat me signals and quantify antibody-mediated phagocytosis in real time, Frodo-labeled CCRF-CEM tumour cells were treated with a concentration range of anti-CD47 or IgG isotype control and co-cultured with BMDM. In the presence of anti-CD47, target cells were rapidly engulfed by the macrophages, with an increase in fluorescent signal being observed, which we can see in the images here at 4 hours. Quantification revealed a kinetic concentration-dependent effect on tumour cell engulfment, with increasing levels of anti-CD47. Conversely, the IgG isotype control had no effect on engulfment. To eliminate the possibility of anti-CD47 treatment resulting in target cell apoptosis and inducing ephagocytosis, phagocytosis and cell health assays were performed. Viable and camptothecin-treated apoptotic CCRF-CEM cells were exposed to anti-CD47 or IgG control in the presence of J774A mouse macrophages. And the results show that anti-CD47 antibody induces phagocytosis of the tumour cells without inducing apoptosis.
The last application I'll be focusing on today is peptide phagocytosis.
Microglia, the resident immune cells of the brain, primarily respond to infection or damage and are involved in the clearance of extracellular amyloid beta, an aggregated peptide that is a pathological hallmark of Alzheimer's disease. This assay enables visualization and analysis of phagocytosis of target material, such as peptides, by immune cells using the Frodo cell labeling kit for Incucyte. Prior to initiating the assay, it's recommended that your experimental design includes replicates of each condition, for example, labeled peptides alone, as well as target and effector co-cultures at each density. So shown here is the protocol for this. So firstly, on day 0, we see the effector cells in a 96-well plate, and these are cultured overnight. The following day, we can then label the target material with the Frodo cell labeling dye. And following the labeling process, we can then add this Frodo label material directly to the plate containing the effector cells. So tips for this protocol and labeling of peptides, we recommend optimizing peptide labeling concentration using Frodo dye that's in a molar excess of the peptide, for example, a range between 50:1 to 5:1. Additionally, during the labeling step, we recommend vortexing and mixing the dye and peptide mixture at room temperature for 4 hours. And you'll notice that this is a slightly longer incubation time than what we recommend for the labeling of cells. Following the labeling of peptides, we recommend quenching any remaining dye using an excess tris buffer. And then the last tip for this is that we recommend using the Frodo orange labeling dye for neuronal cell types, as these cultures can be more sensitive. Here we highlight the ability to efficiently label amyloid beta peptides, accurately quantify engulfment of aggregates, and examine inhibition of microglial phagocytosis using live cell imaging. Axel iPSC microglial precursor cells were seeded into a 96-well plate and differentiated to mature microglia for 2 weeks. Peptides were labelled using the Incucyte Frodo orange cell labelling kit, and aggregates were formed at 37 degrees for 48 hours prior to the assay. The peptides were added to the cells, and phase and fluorescence images were acquired in the Incucyte. And the increase in fluorescence over time can be seen in the images here. We found that the uptake of amyloid beta was concentration dependent, with 100 microgram per mil showing the highest rate of engulfment, with the fluorescence area reaching a plateau by 12 hours. Next, we investigated the inhibition of phagocytosis of aggregated peptides by targeting different mechanisms in BV2 microglia cells. Compounds were used to target cytoskeletal rearrangement, such as cytoclasin D, or block scavenger receptors, such as fucodan or directed monoclonal antibodies. Cytoclasin D and fucodan inhibiting engulfment of amyloid beta aggregates in a concentration-dependent manner. Cytoclasin D showed complete inhibition at the highest concentration. However, for fucodan, only partial inhibition was observed. In the presence of monoclonal antibodies directed against scavenger receptors, inhibition of aggregated amyloid beta phagocytosis was observed, achieving approximately 50% inhibition compared to the IgG isotype control. These data confirm the capability of microglia to engulf disease-associated aggregates and demonstrate how live cell analysis is a powerful method for the functional analysis of immunotherapeutic modulation of phagocytosis. Now to wrap up. The Frodo reagents for Incucyte, in combination with the Incucyte live cell analysis system, provides an integrated kinetic image-based fluorescence solution for researchers to visualize and accurately quantify phagocytosis throughout the entire assay time course. I've provided technical guidance to show you how you can automatically quantify different types of phagocytosis, from bacteria to cells or peptides. And the data shown has demonstrated the applicability of the Incucyte phagocytosis assays across multiple research areas. This functional approach enables high-throughput pharmacological assessment of inhibitors and modulators, while being amenable to screening for novel therapeutic agents. If you'd like any further information on the assays that I've covered today, there are various supporting documents that are available for download from the website, and I've listed a couple of useful links here as well. Thank you for listening to today's webinar,